at this point, you'd be saying, but one of the differences between development and ecological dynamics is that development always sort of produces the same form, whereas ecological dynamics seems to generate a lot of variability. So when NASA started taking animals into space in the 90s, they took vertebrates and invertebrates, and breeding them to perform a variety of experiments, one thing that is quite striking is that they look exactly like animals in the terrestrial environment. So changing the strength of the gravitational force doesn't fundamentally seem to change animal morphology or function. So whereas if it was totally selective, you might think, well, surely there would be some changes if it was a purely selective rather than a programmed development. Is this clear? Maybe gravity is not sufficient. Maybe you have to change many parameters. So what you have to explain then is why development looks algorithmic. Why is there a discipline of development? Why wasn't it always called ecology? And it's because there's something about development that looks different. Right? And this is actually some work that I did with Miguel Fuentes, who's, some of you perhaps know, will talk next week, and a student of ours at Stanford who works in a stem cell lab, who's now at medical school. A little bit more math, not much. So think again about development like this. You have a cell. It can diversify. This is called differentiation in development, or it can replicate. That's called proliferation. So it has a decision at any point. Make a different kind of cell, or make a copy of the same cell. Okay? And you do so at a certain rate, given by these rate constants. Now those rate constants are parameterized. For those who are not really interested in the math, here's the key. You can either have an internal program which tells you what to do at every cell division. So after the first division, I proliferate. Second division, proliferate. Third division, differentiate. Or I can say, wait a minute, what does the other cell say I should do? I can listen to my environment. So there are other cells dividing and proliferating, and they're telling me what their state is through signals. So Pablo could say, you know, I'm in state 4, I think you should be in state 4. So that we call somatic selection. So that there's a selection pressure, meaning something that biases my proliferation and differentiation. So I have two strategies: ignore everybody, or listen to everybody, and come up with a kind of wisdom of crowd solution to what the body should be doing. And the question we were interested in is, development looks algorithmic. It has this feature that the cells seem to do what they want to do, somewhat independently of what the other cells are doing. Okay? Because you don't want a cell in your heart to listen to the cell in your kidney which says be a kidney cell. That might be an error. Whereas in ecological systems, you don't have that degree of internal predestination, right? Because you have a selection dynamic which gives you the correlations with the environment. You listen to the environment. Right? So you do a little bit of maths, and you just make the following assumption. You say, imagine development lives on a manifold, on a surface, and it follows a trajectory. These kinds of illustrations were made popular by someone called Waddington. But sometimes you get perturbed from the trajectory that you want to follow, and you follow another trajectory. Let's call this one a bad trajectory, deleterious. So what development wants to do, either by selection or by programming, is make sure you go this way. Now notice that by selection there's a problem, because if the environment changes, you don't know what's right and wrong, right? It'll push you over here. So you define some functions which tell you how much you deviate from the desired trajectory. So that's all these things are. These say, how much does the development of your process, which is measured in these continuous variables, deviate from the error-free case, r equals 1? I mean, again, I'm just throwing these up to show you that there's a little bit of work here. But conceptually, I think it's quite clear that you're trying to minimize the deviation from a preferred path. And there are two strategies, which I told you before. One is programmed. and one is selective. The selective regime listens to the environment, and the program regime ignores it. Just endogenously specifies the program. And you define this function which tells you the difference between the two strategies. All will become clear with the next picture, I hope. So, here's an error rate, development with no error, development with lots of error, and here's a strength of competition between cells in the body. And what we found is the following. If the error rate is low, so cells, when they divide, they don't make mistakes, okay? And the competition between cells is weak, right? That's when you want selection. So the brain, for example, in the formation of the brain, it chooses a much more selective type approach to consolidating connections. However, when the error rates are high and the competition between cells are high, then you want programs. Because what you don't want is someone who's wrong to tell you what to do. Because you have lots of competition and the error rate is high, and I'm listening to Pablo and he makes a mistake, says, no, you really should be a kidney cell. I really don't want to listen to him. So, here's the point, right? High competition and high error favor program development. So your bodies, somewhat paradoxically and anti-Hamilton, are at the most competitive place on Earth, much more than any ecosystem. So there's this idea by kin selection that all of your cells don't compete. It's the opposite. Your cells will not tolerate deviation from a preferred trajectory. They will outcompete any cell that misbehaves. It's the most competitive ecological arena on Earth of an organism. And again, this one, somewhat counterintuitively, lower competition and lower rates of error give you this Darwinian arena. Okay. So just to recap on this, let me do that. Try to tell you a little bit about niche construction, that the environment doesn't come for free, and that it changes the number of species that you can support in equilibrium. But in fact, developmental variability can be viewed as a form of niche construction. But what makes it look different, and why it has a different discipline associated with it, are factors that are actually ecological: competition, error, and noise. And you can show why development looks programmed versus selective. So we have these bodies by virtue of monopolies, and very strict competition between cells, at fairly low rates of error. That's why bodies exist. You get rid of the monopoly, no bodies, no organisms.
So is there a connection between both program and selection mechanisms?
Yeah, because so it's a continuum. So every cell wants to do something, wants to divide into a successive state, but the rate at which it will divide into that state is going to be dependent on the state of its environment. And that's the standard ecological dynamic. But under certain conditions, it stops listening to its environment, and it puts much more of the information into its own code. And in some limit, it becomes deaf. So if the whole ecosystem were an organism, you could still get diversity and complexity of some kind, but it would all be endogenously driven.
Right, it looks like it might be possible to, if you are programmed, at some point you minimize or you decrease eventually competition and the rate of error, so you might foster the takeover of the selective.
Well, that's absolutely right.
It's like
that's some kind of cyclical.
Right.
No, I think that's absolutely right. In fact, okay, the clearest example of that, by the way, which demonstrates the importance of competition in bodies, is cancer.
Right.
So people say, well, there's no competition between cells. Well, there is, because as soon as cells stop listening to what their other cells are telling them, they take over, and you get a cancerous state. So a cancerous state is actually a shift between the programmed and the selective regimes. So the cell is doing what it wants to do, but with respect to the aggregate, it's now in the wrong stage. And so I think cancer is a very good example that demonstrates that the standard perspective, that the body is not an ecosystem without competition, is wrong. Because all you have to do is lose the ability to listen to other cells, and you take over. So there are instances which are on the path we're describing, but not fully there.
So could you say that organisms that don't get cancer are more obedient in terms of...
Yeah, they're subject to the tyrannical dictates of the monarchy, of the germline. Yeah.
Yeah, but I was thinking in terms of some kind of revolutionary control more or less.
Well, all organisms get cancer, right?
Plants don't.
Well, that's debatable.
Oh, really?
Yeah, and so it's what I mean. One of the problems is metastasis. Is that
Yeah, crazy growth.
right? So you could have local proliferation of tissue in plants, like in galls, which are a sort of induced cancer, right? But they're not metastatic. But the basic phenomenon of the loss of developmental control is probably
Okay. Right.
all right. So again, is this all sort of pretty obvious what's going on here? That you can't, I think for me, you can't take for granted the diversity of an ecosystem. And as a theorist, you need to sort of explain it from the bottom up. And niche construction gives you a way of explaining where the ecosystem came from, ideally, instead of assuming it was there. But then it gives you this other insight that, in fact, you can't assume that there's an environment and an organism, because, in fact, there's a continuity. And that the organism is a special part of the ecosystem which circumscribes its control locally. That's the difference. Otherwise, it's just a continuum. Okay. Now, this approach to development is very simple. There's no genetics in it, for example. Purely ecological modeling. So the question now is, can you do better? Can you go down to genetics too? And this would be more complicated, but I'm going to give you some insights here. So I've told you a little bit about this very simple idea about development, is that you have germlines and they use resources to make soma, stoichiometrically. That's what you do. But where's genetics? Well, most people think development is the interaction between genes, transcription factors, structural genes, to make phenotypes, make proteins and tissues and organs and bodies. So you need that kind of model too. Now, this is Stephen Jay Gould, who's no longer alive, but he was fascinated in this, the implications of the developmental program for evolutionary dynamics. He thought that it had been hugely undervalued, that constraints that come out of the construction process exert a very significant influence on the evolution of life. Okay? So whereas Darwin said, given enough time, the correlation between the organism and the environment will become almost perfect. Gould said, maybe not. Maybe the construction dynamic imposes constraints such that never can be true. And he made a number of statements, I'm going to go through them. Firstly, this has been noted by many people, the so-called sparseness of the morphospace. There are many more forms of life that we can imagine than we observe. Why is that? Why isn't it, over the length of evolutionary time, just filled in continuously? Why isn't there something that is a kind of missing link of morphology? Right? Why is it discrete? The big problem. There are theories that try to address it, we can discuss it during the week, but it's kind of a non-answered question for me. The other one that he was interested in is the so-called violations of phenotypic isotropy. That is, variation doesn't seem to be symmetric. Right? Variation doesn't, imagine a plane where you had 2 dimensions of character. You started here, you might imagine by mutation that you move out radially. So you sample the trait space uniformly. But in fact, it's not true. It's more like this. It's more elliptical. There are favoured dimensions of variation. And he was very interested in that. Because, of course, if there are favoured dimensions of variation developmentally, well then selection can only work on variability, so there will be favoured distributions of species. Over time, there are deviations from what's called uniformitarianism. Charles Lyell, who was also mentioned by one yesterday, is famous for this concept. And this is the concept which says that processes of environmental change are time invariant. So it's like a homogeneous Markov process. Everything we see around us today is the function of forces that we see today. Okay? It's just given enough time, you'll converge to a particular form. But you don't have to invoke differences in time. So the explicit example, the Grand Canyon in North America, you'll say, wow, that must have been a pretty catastrophic event that generated it. And Lyell said, no, you just have rain and you have wind and you have a lot of time. And Darwin appropriated the concept of uniformitarianism for his theory of evolution by natural selection and said, enough time with small changes will give us everything we need. Whereas there were others at the time, the most notable proponent of catastrophism was Cuvier, who said that's not true. So the early Earth was very catastrophic, and that generated lots of variability, and then later it calmed down. And Gould is a catastrophist. Gould said, you know, I don't think it's true. I think evolution, the clock, doesn't run at a constant pace. Sometimes it runs fast, sometimes it runs slow, and so, in consequence, evolution changes its character, what's called its tempo by Simpson. The observation that convergence is much more frequent than we assume. Does everyone know that convergence is like different lineages having the same structures, like wings, or the vermiform structure of snakes and worms? They didn't inherit that structure, they converged. And this is a slightly more recent and slightly more technical remark, and that is that all of this stuff is guided by what are called regulatory genes rather than structural ones. And I'll talk more about this in a second. I mean, most of you are biologists here, so this means something to you when I say regulatory versus structural gene, or not? No, I mean, I don't know. Just tell me, I'm not offended, don't be shy. I mean, it's fine, right? If you don't know, it's fine. So, just to sort of calm things down a little bit, the clearest example of the kind of Gouldian worldview in practice is in the TV series Star Trek, where whichever planet Captain Kirk goes to, he can always find someone to have sex with. It's actually a very interesting characteristic of Captain Kirk, right? And because they're all basically humanoid, right? They're all bipedal, they're all vertebrates. They all have 2 eyes and 2 legs and 2 arms, with a few exceptions. Now, I've shown this slide before at talks, and I'll tell you that I will buy 2 beers for anyone who can name all of these species, and I'll buy 1 beer for anyone who can name these. When I give this talk in Santa Fe, one of the girls in the audience could name 6. Yeah. It shows you how we spend our time, right? But I'm sure how many, can you name this crew? I'm very sort of pessimistic about the Chilean contribution to the Star Trek universe. And so, but can anyone name even a single species on this slide?
But it's not easy to see them.
What do you mean? Maybe there's too much light.
That's stronger.
No, this is a first. You guys can't even name 1. This is just horrifying. So,
a Krakauer.
Yeah. No. Yeah. Well, I'm going to tell you, there are very few exceptions to the rule in Star Trek that morphologies, wherever you find them in the universe, are fundamentally anthropoid. I think it's kind of interesting, right? And it's something to do with empathy, presumably. But there are this one, the Davinian neural parasite, the tribble. I mean, most people know tribbles, for Christ's sake, and the horta. I want to tell you guys that at least 50% of the audience in Santa Fe knows those. You need to go and rewatch Star Trek. And there are 500 species in the Star Trek universe, and I highlighted the ones that you just saw. And many of them have this anthropoid property. You will look horrified. You're probably all reading Proust rather than watching Star Trek, which is a little bit intimidating. All right. So, let's go back to something more serious. The question is, what makes the Gould doctrine, or the common structure of the Star Trek universe, likely? So I'm now going to tell you that maybe it's not as stupid as it seems. Okay? So this is how you might write down, a little bit more formally, a regulatory network in genetics. And there are many ways to do it. Those of you who come from mathematics and physics and engineering will recognize this as essentially a perceptron, or a single layer neural network. It's just linear algebra that I'm showing now. So what you have is you have transcription factors. These are the genes that turn other genes on and off. And they bind to the promoters of structural genes. So here you have a number of transcription factors, and here you have a number of structural genes, and they bind to them with different affinities. And if they bind very strongly, the gene gets turned on. If they bind weakly, it doesn't get turned on. And so you can write down very simple, you can generate a random matrix for development. You'll generate something like an Erdős-Rényi graph. You just assume that there's a constant probability of it being occupied. And then you just multiply the developmental matrix, right, by the transcription factor vector. You push it through a nonlinear function, and you derive a phenotype vector. Okay. So matrix multiplication with a nonlinearity. So income, transcription factors, development, outcome, phenotype, and they're both represented in this binary vector space for convenience. So you can imagine what I just told you, what would be called a single projection, a mapping from genotype to phenotype, which is just a mapping from one vector to another. Okay? Or you could actually feed the output back into the input. This is called a recurrent design. Okay? This requires that the developmental matrix is square, the dimensions of the vectors are the same. Or a recurrent random projection where the developmental matrix is no longer time invariant. It's a non-homogeneous Markov process. You care about such things. So you can write down models, and these have been extensively explored now by systems biologists, geneticists. One of my colleagues, Andreas Wagner, has worked a lot on these models. It's perhaps the most popular model for exploring developmental dynamics. What are the consequences of writing down these models? Now, a number of slides, but the basic point is interesting and kind of obvious. I'm going to define a regulatory dimension. The regulatory dimension, right, is just, if you like, the number of bits in the transcription factor vector. So I told you you had this transcription factor vector. It has ones and zeros and so forth in it. Okay? And the number of elements there, let's call that R. So that's the regulatory dimension. And as you increase the regulatory dimension, the number of possible phenotypes, when you multiply that by a matrix, imagine you multiply it by the diagonal matrix, is going to be 2 raised to the power of R, because it's a binary vector. The total number of vectors is 2 to the R. And so this is the number of phenotypes, vectors that you could generate as you increase the number of bits in the transcription factor vector. But if you actually look at the number of vectors that you generate, right, it's much, much less. Okay? And so this is the regulatory dimension, divided by the total possible. The observed divided by the possible 2 to the R. And what you see is, as you increase the regulatory dimension, the percentage of phenotypes that are visible, expressed, drops systematically. Okay?
So when you say visible or expressed, are you talking that you have data? That you somehow...
So what you do is you look at this model and you calculate, you enumerate over all, let's say, 2 to the R input bit strings. You throw them through that simple mapping, and you get out phenotypes which are binary vectors, and you count them.
Okay.
You know if the developmental matrix was diagonal, and right, the square matrix, that they would be equal. You have the same number of phenotypes as genetic configurations. But it's not true, right, for non-diagonal matrices. And it turns out that for non-diagonal matrices, you have very few phenotypes. And that you get fewer and fewer and fewer the higher the dimension. Get that one. That's the first critical point. In fact, and this is the only power law in the talk, you can define a neutrality level. And what neutrality is, it shows you basically, this is the mapping, these are one-to-one mappings. The number of a unique genotype generates a unique phenotype. Over here, right, you have many, many genotypes that make the same phenotype. Okay? So on the left-hand side, one genotype, genotype 1, maps to phenotype 1. Whereas on the right-hand side, these are all these cases where you have multiple genotypes, and they all map onto the same phenotype. So you'll notice over here that you have cases where you have over 1,000 genotypes all mapping to the same phenotype. That's the consequence of the developmental map. It compresses the image of the pre-image. Right? It goes from a volume in this n-dimensional sphere in your vector space, it's mapping between two spaces. It goes from a big one to a little one. And this is just illustrating the point further. And what's interesting about it, look at the right here, this is a phenotypic network. Each node here is a vector in phenotype space, and the radius, right, is proportional to the log of the frequency. So the more phenotypes, the bigger the circle. And what you notice in this space, it's not a genetic space, it's a phenotypic space, is that most of the most common phenotypes are neighbors. So a very interesting property of these developmental maps is that most of the phenotypes that you see in the world should look like each other.
What does it mean to be a neighbor there?
Because you can define phenotypes in the binary vector space, it's Hamming distance. And so two nodes are connected when they differ by 1 bit. Jeff?
Do the phenotypes look alike because of developmental reasons, or is there exposed to similar environments?
There's no selection here. Purely because of the properties of pleiotropy and epistasis in the developmental matrix. There's no selection. Yeah, there's nothing.
Those are just visible phenotypes?
Mm.
Those are just
Visible.
visible phenotypes?
Visible.
Yeah.
Yeah.
Visible
Because there are many.
Yeah,
Oh, of course, because most of them are visible. Yeah, these are visible phenotypes. So, again, I want to emphasize, we're trying to answer some of those remarks that Stephen Jay Gould made about biases in the developmental plans that are selection-free, that come from constraints. And this is just an illustration of the principle that if you do any projection through any dynamical system, and this is a very simple one, and I'll show you it gets much worse, you have to get what Stephen Jay Gould was suggesting. And for those of you who don't know about evolution, the common assumption in evolutionary dynamics is that this is not true. That all genotypes can be accessed. The only constraint is the probability of you're more likely to move to something more similar to you. But there's no fundamental constraint in terms of the occupancy of morphospace. But as soon as you put a developmental plan in, it has to be true. And it's true in a way that's very similar to what Gould intuited. That's just, yeah.
Sorry, one more question. So do the dynamics of modeling constraints are fundamentally different than the dynamics of modeling selection in the conventional sense?
Absolutely, absolutely.
They're fundamentally different.
They're fundamentally different, yeah. Yeah. Is this clear to people? I mean, just to give you a certain intuition, is that when genes are interacting, when I interact with Pablo and Pablo interacts with Jessica and so on, that starts imposing constraints on our degrees of freedom. So before we could move anywhere we wanted, now we can't. So any model that you create is going to have this restriction effect. What's interesting about it is it's restricted in a way that's structured. The common things are like each other. That didn't have to be true if you hadn't written down the mathematical model.
So, I was wondering if you can have the same effect, but from a different principle, just because of you increase your number of transcription factors,
Yeah.
and if you put some noise,
Yes.